Hello everyone and welcome to Lab 11, the topic of which is oxygen requirements. As the name of this lab suggests, we are going to be exploring the different oxygen requirements of categories of microbes by cultivating them in a special type of media that will allow us to visualize the specific part of a tube that the microbes grow in and thereby make conclusions about what its oxygen requirements are. There are 5 different groups of microbes that can be classified based upon their relationship to oxygen. The first one that you see in this table is called the obligate aerobe. Now, sometimes they're just called aerobes, but I've also heard them called obligate aerobes, but they mean the same thing. An obligate aerobe is an organism that requires the presence of oxygen in order to carry out aerobic cellular respiration. Obligate anaerobes are organisms that require the absence of oxygen, which may sound sort of counterintuitive to us as organisms that require oxygen to live. We are obligate aerobes, but there are microbes out there that are obligate anaerobes and require the absence of oxygen. And we'll talk about how that can be possible in just a second. Aerotolerant anaerobes do not perform cellular respiration, and therefore they do not use oxygen, but they tolerate its presence and are not harmed by it. Facultative anaerobes are capable of growing either in the presence or absence of oxygen. When oxygen is available, they can perform aerobic cellular respiration, and when it is not available, they can perform fermentation, although they grow more efficiently in the presence of oxygen, given that cellular respiration is a more efficient process. And then last but not least, there are microaerophiles, which require limited amounts of oxygen, and I like to describe them as picky because they need not too much but not too little oxygen, somewhere in between. So how can it be the case that there are organisms that are harmed by oxygen and cannot live in its presence, the obligate anaerobes? Well, this is actually the case for all of us. All organisms are harmed by oxygen because, as a natural byproduct of our metabolic processes, oxygen is transformed in very small amounts into this compound called the superoxide anion, which is toxic because it is highly unstable. Organisms are only able to survive and tolerate the presence of oxygen because of 2 enzymes that have evolved to deal with the superoxide anion problem, one of which is called superoxide dismutase, which is capable of taking 2 separate superoxide anions, combining them with protons, and transforming these things into oxygen and hydrogen peroxide, H2O2. Now we've still got a problem here because hydrogen peroxide is also toxic to living organisms, and therefore a second enzyme called catalase is needed in this process, which breaks down the hydrogen peroxide into harmless water and more oxygen. So this explains why it can be the case that there are organisms that are harmed by oxygen. Obligate anaerobes do not synthesize superoxide dismutase or catalase, and therefore they are not able to deal with the toxic forms of oxygen that all living things produce as a part of their metabolic processes. So we are going to look at these 5 different categories of microbes in this lab, and we're going to explore the way that they grow in tube media by carrying out what are called shake tube inoculations. The purpose of a shake tube inoculation is to inoculate a solid media with an inoculum that gets evenly distributed throughout the media, and then it is allowed to solidify so that the microbes remain suspended in that solid media, and you can examine the area of the tube in which they grow. The type of media that is used in this lab is called brain-heart infusion agar, which is a formulation of agar that is highly dense in nutrients. It contains the extracts of cow or pig hearts and brains, and this allows for the growth of fastidious microorganisms, which are organisms that have highly complex growth requirements that are kind of difficult to pin down in the laboratory setting. So we start with solid brain-heart infusion agar tubes. But in order to inoculate these guys, they need to be liquefied. And so the tubes are exposed to a water bath at 100 degrees Celsius, which causes the solid agar to melt. After that, the tubes need to be cooled down to a temperature that won't kill the inoculum, and that temperature is 45 degrees Celsius. The tubes need to be cooled to a point where they're cool enough not to kill the bacteria, but they're not so cool that the agar is going to re-solidify. And so 45 degrees Celsius is sitting at that perfect middle point. Once the tubes are cooled, then we add 2 drops of each of 6 stock cultures to the tubes. So there are 6 separate microorganisms that are worked with in this lab. We have Staphylococcus aureus, Corynebacterium xerosis, Enterococcus faecalis, Saccharomyces cerevisiae, Aspergillus niger, and Clostridium sporogenes. And by the time you look at the results for this lab, you should be able to tell what oxygen requirement is assigned to each of these species. So there are 6 species that are inoculated in this manner. After the drops of stock culture are added to each of the liquefied agar tubes, then the tubes are capped and twirled between the palms in order to mix them and draw the inoculum all the way to the bottom of the tube so that it is evenly distributed. And this is sort of the namesake of the shake tube inoculation. You are twirling them or shaking them in order to distribute the inoculum and mix it throughout the entire media. After this, you want the tubes to immediately solidify because you want those microbes to remain suspended in the media when it is solid. So the tubes will be placed upright in an ice water bath in order to rapidly solidify that agar again. So let's take a look at what this looks like in the lab. Alrighty, so hopefully you remember that the first step in this process is to heat up our BHI tubes so that we can liquefy the agar. We have here a boiling water bath that is set to 100 degrees Celsius boiling temperature, and inside we have our BHI tubes. Right here we can see that they are liquid, but we need to cool these down to 45 degrees Celsius, and so I'm going to place them in a cool water bath in order to do that. The reason being, they cannot be too hot such that they kill the microbes, so we need to cool them off. Once they are cool enough as to not kill the microbes, but not so cool that they have re-solidified, then we will inoculate them with our species. Okay, so our tubes have now cooled, and we're ready to inoculate them. We're doing the shake tube inoculation technique, which means that we're going to be putting drops of inoculum into the liquid tubes, then twirling them to distribute the inoculum, and then plunging them into this ice bath next to me in order to re-solidify the media. So here we have some sterile disposable pipettes that are going to be used for the dropping procedure. Here's my pipette. We're going to start with Staphylococcus aureus. I'm going to go in, pick up a small amount of inoculum, two drops in the tube, and now I'm twirling this tube to distribute the inoculum, and finally plunging it into the ice bath in order to rapidly solidify that BHI agar so that the inoculum remains distributed throughout the media when it becomes solid. Alrighty, all 6 of our BHI tubes have been properly inoculated and thrown in the ice bath, so they are solid again, as you can see. And now all 6 of these are going to be incubated in our 37 degrees Celsius incubator for 48 hours, and then we will look at the results. Now that you've seen these techniques carried out, let's talk about the interpretation of the results. When you look at the results, what you're going to be examining is where the microbes are growing in the tube. Each of the different classes of microbes has a different oxygen requirement and therefore will grow in a different pattern in the tube. Obligate aerobes, as we know, require the presence of oxygen, and therefore they can only grow towards the top of the tube where there is the interface between the solid media and the air. Facultative anaerobes, as we learned, are capable of growing with or without oxygen, but they grow more efficiently with oxygen. So a typical facultative anaerobe will have growth distributed throughout the tube, but the growth will be more highly concentrated at the interface where oxygen is present. Obligate anaerobes require the absence of oxygen, and so therefore these sorts of organisms can only grow below a certain point where dissolved oxygen is low enough that it is no longer toxic to them. Aerotolerant anaerobes do not use oxygen and therefore get no benefit from it, but they tolerate its presence, and so aerotolerant organisms are expected to grow evenly throughout the tube. And then finally, microaerophiles, the picky ones, they require a fixed amount of oxygen that's not too much, not too little, and so these guys will grow in a distinct stripe somewhere in the middle of the tube where there is just the right amount of dissolved oxygen.